want to do is direct this talk to the students and postdocs in the audience. And what I want to do is tell you a few stories, scientific stories from my lab, but use those to illustrate lessons that I've learned during my career development. So I'm going to first summarize the key lessons that I've learned. And those are to choose a problem in biology as the basis of your research program, one that you're really excited about. Second, to exploit the experimental advantages of the system. That's a great thing in flies because you can do it so readily. And third, in this Drosophila community, to be a part of this extraordinary community. So let me give you a little bit of history. When I started my lab in 1987, really there was an explosion happening in the developmental biology field and also the cell cycle field. So where we were at in 1987 is that many of the regulatory genes for establishing pattern and cell identity had been identified. And those genes were in the process of being cloned. So we were beginning to understand what kind of proteins were the key developmental regulators, what they controlled during embryogenesis, and where they were expressed. So it was a pivotal point in the developmental biology field. It was also a pivotal point in the cell cycle field, because what had happened is there was a convergence of genetic approaches from the yeasts together with biochemical approaches, largely from extracts from Xenopus oocytes and early embryos. And what those studies had done was to identify critical regulators of the cell cycle and cell division, namely the essential role of these cyclin-dependent kinases, or CDKs, that when complexed with cyclins would drive transition through the cell cycle. And the first of those being the CDK1 cyclin B complex conserved from yeast to humans, which is essential for driving the onset of mitosis. So as I was thinking about setting up an independent research program, I wondered whether it would make sense to work at the interface of these fields, that to really understand development, it was going to be important to understand how cells became determined and differentiated, but also to understand how the right number of cells got made and positioned in the right tissues. And so when I started my lab, that's what I looked like. I decided that the umbrella theme for our research would be how is cell division coordinated with development, and of course we looked at this in Drosophila. Now this turned out to be particularly interesting in Drosophila because in addition to the established canonical cell cycle, in which there's a DNA synthesis phase and a mitotic phase of segregating the chromosomes punctuated by these gap phases during which gene expression and growth take place. What became apparent is that Drosophila extensively used variations of this cell cycle to achieve particular developmental strategies. And I've already introduced you and alluded to one of these, which is the early embryonic division cycle, which is an accelerated cycle, a stripped-down cycle, where there's an oscillation, a rapid oscillation between DNA synthesis and mitosis without any transcription or taking time for any growth. And this is made possible by the stockpiling of the embryo with necessary maternal components. Another variant cell cycle is one that ultimately came to be called the Endo cycle. This is a cycle in which there's a synthesis phase and a gap phase when gene expression and growth takes place, but it's completely dissociated from mitosis and cell division. And so you can see that this increases the DNA content and the ploidy of the cells. So my first two students began working, one of them on the SM cycles and one of them on the SG cycles. And Albert Smith was not only my first student, but he was also the first person to join the lab. And he did a lot of the early work establishing the developmental control of the SG endocycle and the parameters of it. So what I want to do tonight is tell you 3 vignettes that tie into these variant cell cycles. The first thing I want to talk about is what we've learned about the regulation of the oocyte-to-embryo transition. So how is the regulation that take place to actually set up the embryo to begin these SM cycles? And then I want to turn to the endocycle and tell you what we've learned about how you can make big, really big cells. And then finally, I want to tell you about how we've exploited that to learn some interesting things about regulation of replication for progression. So let's begin at the beginning, which is the oocyte-to-embryo transition. So this is a massive transition. I assert this is the most important step in development. Of course, I know that there might be some disagreement about that, but it involves taking this differentiated oocyte, which is arrested in metaphase of meiosis 1, and causing that to complete meiosis to reset to be able to undergo mitosis during embryogenesis, and then to have this profound change in potency to go from being a differentiated cell to a totipotent cell. And what's remarkable about this is in almost every animal, this transition occurs in the absence of any transcription. And that means that all of these myriad of changes have to be regulated by changes in mRNA translation and protein stability. This is a great problem to study in Drosophila for a variety of reasons. One of them, of course, genetics being one, another being that these stages of meiotic progression through oocyte maturation to the metaphase 1 arresting the mature oocyte are all morphologically distinct. And so that means that biochemically you can isolate sufficient amounts of each of these stages to do biochemistry. Another is that the resetting for this transition happens as the mature oocyte transitions through the oviduct, and that in a process that I'll term egg activation. And although that usually is coincident with fertilization, it's actually not dependent on fertilization. So egg activation will trigger all these changes in protein stability, mRNA translation, all the cell cycle changes. And we can study all of them without any confounding effects from contribution from the zygotic genome itself. And it also means that we can study we can actually make this happen in vitro, as you'll see. So when we started this project in the lab, we decided to do a very direct visual screen to look for mutants that were defective, either in the completion of meiosis or in the restart of SM cycles in embryogenesis. And the community helped us very much to get started because Trudy Schupbach, Christine Nusslein-Volhard, Eric Wiechhaus, and Dawson Moeller let us screen their maternal effect lethal collections. And we screened them simply by DAPI staining the eggs laid by these mutant mothers and looking for defects in the oocyte-to-embryo transition. And so here's a phenotype that came out of the screen that was very striking. On the top, you can see a wild-type embryo filling up rapidly with these diploid nuclei as it goes through these SM cycles. And you can see the phenotype we decided to pursue on the bottom. And what's going wrong here is that meiosis is completed. And all those meiotic products and the male pronucleus, if it's fertilized, all start that first S phase, but then they cannot do the first mitosis of embryogenesis. So they just continue to generate these larger and larger polyploid nuclei. So when we did the genetics, the alleles that we had isolated all fell into 3 complementation groups. We named 2 of them Pangu and plutonium, or Plu, and there was a 3rd gene that had already been identified in David Glover's lab called giant nucleus, or GNU. And we found that all three of these genes genetically interacted, which was suggestive that they all work together on the same pathway. So the naming of giant nucleus, I think, is obvious. It's too long of a story to tell you how we named Pangu, but I'll tell you why we named plutonium. We named it plutonium because it has a giant nucleus and it was lethal. So we were able to really sort out what seemed to be the primary defect responsible for the phenotype that we were seeing. And that is, as I mentioned, what happens in eggs laid by these mutant mothers is they start the first S phase, but then they can't do the first mitosis. And we showed that was because they had inadequate amounts of cyclin B protein, because Pangu, plutonium, and GNU were all required for the translation of cyclin B protein. And we could completely account for everything by this. However, in parallel, Howard Lipschitz's lab was looking at a later event in embryogenesis, the handover from maternal to zygotic control of development. And what they found is that Pangu, plutonium, and GNU were required for the translation of SMOG protein. Yes, it's SMOG named after the dragon. And SMOG is required later in embryogenesis for deadenylation of maternal messages and their degradation. So it seemed like everything we could find wrong with these mutants could be explained by them being required in a positive way to promote translation of cyclin B and SMOG. Now, an alternative approach that we took to understand the oocyte embryo transition was to do some biochemistry. And as I said, we could isolate all of these stages, including activated eggs, and analyze the changes that occurred. And so what Eva Kronia in my lab did was she analyzed, using quantitative proteomics, she analyzed all the changes in protein levels during oocyte maturation and during egg activation. In collaboration with David Bartel's lab, she also analyzed all the changes in mRNA translation. And also, in collaboration with Bartel's lab, she analyzed all the changes in polyadenylation of these mRNAs. And so I'm just going to summarize her results for mRNA translation. If we compare for each of the mRNAs that are present in the oocyte and the embryo, and as I've asserted, there's no change in mRNA levels through oocyte maturation and egg activation. If we compare the efficiency of translation of those between mature oocytes and activated eggs, what we see is there is profound changes in translation. And so it's not the case that the mature oocyte is this quiescent, in a quiescent state with regard to translation. There's extensive mRNA translation going on in the mature oocyte. And at egg activation, there's dramatic remodeling. So what happens is out of these 6,000 RNAs from 6,000 different genes in the mature oocyte, at egg activation, about 500 of them become repressed for translation, and about 1,000 of them become activated. So it's a really dramatic, profound change. And now having this catalog of all these changes in mRNA, what we decided is we should look at Pangu mutants because maybe there would be a handful more targets. That's the way I was thinking about it, that maybe we would find a few more targets of Pangu that might reveal additional functions. And so we did that, and we were shocked at the result. I don't know if I looked like this, I certainly felt like this, because what we found is that of the 1,000 mRNAs that become translationally activated, 85% of them are dependent on Pangu activity. And not only that, but of the 500 or so that become repressed, 90% of them were dependent on PANGU activity. So now we had to think about PANGU in a completely different way as really this global regulator of mRNA translation. And the genetics suggested that it was a global regulator of mRNA translation, specifically at this developmental window between the oocyte and the embryo. So it seemed like this might break pretty quickly because when we cloned the genes, what we found is that PANGU should be a serine threonine kinase. And so it seemed like it might all unfold in a pretty straightforward way, but science doesn't always work like that. And what ensued were several years of extreme frustration where it had to be a kinase. The alleles told us it had to be a kinase. The null alleles were in the catalytic domain. It had to be a kinase, but no matter what we did, we could not get this to have any kinase activity. So we really fought with it, and in the end, it had a very nice biological explanation. And that was the reason that these genes genetically interacted is their protein products all form a complex. PANGU is the catalytic kinase subunit, but it's completely dead unless it's complexed with the other two, the plu and the GNU proteins, which are activating subunits of the kinase. So here's what we knew then. We had identified a kinase complex. That kinase complex is a global regulator of maternal mRNA translation, both for things that are activated and things that are repressed at the transition from oocyte to embryo. And so this raised really two essential questions. One is, how is PANGU developmentally regulated? And the second is, how does it control translation of hundreds of mRNAs? And we feel like we've really cracked the developmental regulatory question. And it's very exquisite control that I'll tell you about. And I'll tell you where we've gotten to in terms of understanding the mechanism by which it controls translation. So we began by asking when these proteins are present. So these are Western blots of the stages through oocyte maturation, stage 14 being the mature oocytes. And we found the proteins of the complex increase, and they're all there in mature oocytes. But remember, the kinase is dead unless the three proteins are physically associated. And what we found in mature oocytes is that PANGU and plu are in a complex, but GNU, although present, is not bound to them. So the question is, how does it get activated? And fortunately, we could activate mature oocytes in vitro by hypotonically swelling them and use that as a biochemical system to ask when the kinase becomes active. And as our readout for when the kinase becomes active, we could look for the appearance of SMOG protein, which is completely dependent for its translation on PANGU activity. So let me walk you quickly through these Western blots where we're looking at time points after egg activation, and we're identifying when PANGU is active by the appearance of SMOG protein. And so basically what we found is that at this time point, PANGU becomes active, and this corresponded with when meiosis was completed by cytology. It also corresponds with when you see a drop in the mitotic cyclin levels. I want you to also notice it corresponds with this real change in mobility of the GNU protein, and we showed that's because GNU becomes dephosphorylated. So here's how this elegant developmental control of this kinase works. Everything's there in the mature oocyte. PANGU and plu are together. GNU is phosphorylated by cyclin B-CDK. And in this phosphorylated state, it is unable to bind to PANGU and plu, so the kinase is inactive. But at egg activation, As the meiotic arrest is released and cyclin B is degraded, we see that GNU becomes dephosphorylated. We see it form a complex with PANGU and PLU to make active kinase, which then regulates the translation of maternal mRNAs. And what's interesting is then GNU becomes degraded in a PANGU-dependent manner. So this mechanism restricts the activity of PANGU to a very narrow developmental window of less than an hour, and the onset is dictated by the completion of meiosis, and then the turning off is dictated by having formed an active PANGU kinase complex. That's the developmental control. So the question is, how does it control translation? This is the open question that needs to be solved. There likely are many mechanisms. The way we've gone after it is to do unbiased biochemical screens to identify PANGU substrates. And I'll tell you the results from a recent one where what we found is PANGU phosphorylates many components of the translation machinery, including a number of translational repressors. And for 2 of those, we've shown that that correlates with blocking the repressor's activity, which would then permit activation of maternal mRNA translation. So an important question is, what does GNU do? What is it doing when it's phosphorylated in the mature oocyte? Does it have activities independent of PANGU and PLU? These are questions being addressed by Amir Aviles-Pagan, a student in the lab, and please see his poster for details. Let me also say that what came out of our proteomics screen was a recognition of how important a change in redox is in regulating the oocyte-to-embryo transition. And so please see the poster by postdoc in the lab, Boriana Petrova, on her genetics and biochemistry, defining these essential redox changes, both for the completion of meiosis and for egg activation. So what did I learn as a developing scientist from this project? I learned that you need diverse approaches to solve biological problems. We really had to move beyond genetics to do biochemistry and omics to understand this. I learned that you have to be resilient and keep pushing. We really fought to get the PANGU to be a functional kinase. And I learned, importantly, that students and postdocs are resourceful, and their willingness to tackle new approaches in a fearless way and to ferret out people to help them with those new technology was really critical for the success of this. As part of that, I learned that collaborations are powerful, and they permit combined expertises to be applied in a synergistic way. So we collaborated to do the quantitative proteomics with Jeroen Krigsveld at the EMBL. As I mentioned, all the translatome stuff we did was with David Bartel. The PANGU substrate screens we did together with Mark Kirshner early on, and then later with Sebastian Laredo, and Ben Turk at Yale helped us with substrate specificity. And our collaboration with Howard Lipschitz on smog showed us this additional function of PANGU. So let me turn now to the second vignette, which comes out of the Endo cycle. And so again, this variant cell cycle, in which DNA replication happens in an unlinked way to cell division, leads to increased DNA content, or what we can call polyploidization. And you're familiar with this undoubtedly from these beautiful salivary gland Polytene chromosomes, where each of these chromosome arms has about 2,000 copies of DNA duplexes held and registered to give these beautiful banding patterns. But what you may not be aware of is there are somatic polyploid cells throughout the plant and animal kingdom, and what they are associated with is large cells. So this is true in humans as well. So for example, these giant trophoblast cells in the placenta. There's a layer of polyploid cells in the outer layer of your skin. There's the way megakaryocytes become large is by polyploidization. And in plants, the distribution of polyploid to diploid cells can control architecture of tissues like these giant cells in the sepal. So the second vignette then is, how do you grow a big cell? And by this, I don't mean some measly, little 1.5 to 2-fold change that you can get out of growing out of a G phase. I mean orders of magnitude. So I'm showing you here, for example, some glial cells where their boundaries are outlined in the brain lobes. And the surface area of these glial cells is 15,000 micrometers squared. And so these cells are 10,000 times bigger than a diploid epithelial cell. So how do you grow a large cell like this, and why would that ever be necessary as a developmental strategy? The thing to think about here is the way we usually think about tissue growth is by increasing number of cells via proliferation. But what I'm arguing is there are developmental contexts where it may be important to have very large cells. And really throughout biology, cell size scales with DNA content. So the most common way to accomplish that is by polyploidization. So I'm going to tell you about work in the larval nervous system, where we have found that at least 2 types of glia use polyploidization as the strategy to control their growth. The ones we've really established to do that are the subparaneural glia and the wrapping glia in the PNS. And tonight I'll tell you about work on the subparaneural glia. So these subparaneural glia, which from now on I'm going to call SPG, are present throughout the larval nervous system in the brain lobes, in the ventral cord, and in the peripheral nervous system. And they form the outer layer. And a number of labs have shown that they're essential for the blood-brain barrier. Work from the Gall lab, the Heberlin lab, and the Volk lab have shown that they are required to control, act as a barrier to control entry of metabolites from the hemolymph. And work from the Brand lab and the Gould lab has shown that they also have a niche capacity to control neuroblast proliferation. So these are flat cells. I'm just sorry if I keep making you deaf by hitting the microphone. These are flat cells that you can really think of as a layer of saran wrap surrounding the nervous system. And they're joined all around their periphery by these septate junctions. And those septate junctions are essential for a functional blood-brain barrier. So we started looking at the SPG, and we could do that by using specific GAL4 drivers. So in the slides I'm going to be showing you only the nuclei of the SPG are labeled and the membrane boundaries of the SPG. And what we found, strikingly, is that the number of SPG cells didn't change during larval development at all. There's 13 cells in the brain, despite the fact that there are these massive increases in the size of the nervous system. But what we were able to show is that these cells were increasing in size, and that was associated with these large changes in their ploidy or DNA content. Now we could test the function because we could specifically block the ability of these SPG cells to replicate their DNA, and then ask the consequence. And of course the key function of these cells is to regulate the blood-brain barrier. So when we did that, what we found is if we didn't allow these cells to grow bigger by replicating their DNA and increasing their ploidy, the blood-brain barrier failed. And you can see that here, but this is an experiment where we've injected a fluorescent dextran into the body cavity in this control where the blood-brain barrier is functional. The dye doesn't enter the brain. But if we prevent these cells from increasing in size, the blood-brain barrier fails. And so here's what we think is happening. You need to focus on the clothing here. Normally, these SPG saran wrap layer increases in size as the underlying neuronal mass is increasing. But if we prevent that increase in size, then the clothing gets ripped. And indeed, if we look at the septate junctions, we find that if we block DNA replication, the septate junctions all break, coincident with the loss of the blood-brain barrier. So what we think we're seeing here is a paradigm for developmental situations in which increasing cell size is critical for making a barrier. So the way we think about it is that in cases where you need a tissue layer to act as a barrier, like these trophoblast giant cells in the placenta, which make a barrier between the maternal and fetal compartments, if cell division happens, at least transient, there's going to be a break in the barrier and potential leakage. And so allowing the cells to grow but not to undergo cell division is a way to solve this problem. Now, it's interesting because there is regulation across the tissue layers. So when we asked what happened in a tumor mutant where the neuroblast divisions are unregulated, so the mass of the underlying neuronal tissue is markedly increased, we were surprised to find that the blood-brain barrier was fine. But what we saw is that in this case, the SPG had responded to the increased neuronal mass by increasing their ploidy and size and thereby preserving the blood-brain barrier. And what this indicates is that there's signaling between the neuronal mass to really affect the ploidy and size of the SPG. Now, those of you looking carefully at these SPG in the brain lobes may have noticed that there's two types of these cells. Again, we've got the septate junctions outlined in green. There are cells that only have one nucleus, and there are cells that have multiple nuclei. And, you know, I've been emphasizing this Endo cycle where there are no aspects of mitosis or cell division because this is what's used extensively in differentiated tissues in Drosophila to control cell size. But there are other variant cell cycles that can also give rise to polyploidy. And I'm putting several of those under the heading of endomitosis. I know this is not the classical definition of endomitosis, but I think it's useful to group variant cell cycles that do some aspects of mitosis but not cytokinesis under the heading of endomitosis. And the particular form of endomitosis that we're seeing in these SPGs is in a situation where they undergo chromosome segregation and nuclear division, but not cytokinesis. And so that produces these multinucleate cells. So this is, and so about 70% of the SPG in the brain, in fact, are multinucleate. This is under developmental control because what we've established is that in embryogenesis, nearly all or all of the SPG enter the endocycle. But then between the first and the second larval instar stages, in the majority of SPG cells in the brain, mitosis is reinstated. And that's associated with activation of cyclin CDK1 mitotic cyclins under the control of the string phosphatase. It's also regulated by notch signaling. So notch signaling is required both to retain the fraction of SPG that are going to stay in the endocycle and also to limit the extent of nuclear division in those SPG that enter endomitosis. So why do this? Well, what we've learned so far is the outcomes of endomitosis and the endocycle are different. That the endomitotic cells can attain a higher ploidy and a larger size. And we are able to now perturb the ratio between endocycling and endomitotic cells by changing notch signaling or string activity. And what those experiments show is that, in fact, both endomitotic and endocycling cells are critical for the blood-brain barrier. So why do endomitosis? How is endomitosis different from the endocycle? For the way we're going about answering those questions, I'll refer you to Jessica von Stettina's talk on Friday night, where you can hear what's new on this story. What did I learn from the second vignette? I learned to keep your eyes open for interesting biology. We saw by Dappy's staining that there clearly had to be polyploid cells in the nervous system, and decided to go for it. We saw that there was something very different about these SPG cells in the brain lobes, and that they were multinucleate, and to track that down. So I learned from that not to be afraid to go into a new field, even something as different as neurobiology. And here again, I was able to really learn from my students and postdocs, and they took the lead in learning this new field and helping to teach it to me and helping us all to move into it. I also, this has been emphasized several times earlier this evening, but it's important to reiterate it, these community resources that our colleagues so generously generated as toolkits for us were critical for our work here. The GAL4 driver lines allowed us to label the glial cells and to track them. Those initially were started by work from Andrea Brand and Norbert Peramon, and of course many people in the community have continued to add to those collections. We also needed and used the RNAi lines from both the TRIP and the VDRC collections, work that was led by Elizabeth Perkins, Norbert Peramon, Christina Kellerman, and Barry Dixon. Okay, this has left us with lots of interesting questions. I've already mentioned some of these. Why are the growth properties of endomitotic cells different than endocycling cells? How is SPG ploidy controlled by neuronal mass? And thinking about how this might relate to the vertebrate system, are those endothelial cells inside the vasculature that control the blood-brain barrier in vertebrates via tight junctions that are parallel to these septate junctions? Are those cells maintaining their size by polyploidization? All right, vignette number 3. This also ties into the endocycle. And this is a situation where we thought about how could we use this variant cell cycle to address a problem, a very important problem in biology that was not tractable in other systems. And the problem we realized we could go after was to really begin to understand how replication fork progression is regulated. And so we looked at two questions. How can replication forks be inhibited? And what happens when replication forks collide? So let me remind you about a fact about this SG cycle. And that is that during the S phase of the SG cycle in Drosophila, there's not necessarily a uniform integral doubling of the genome. So most of the tissues that have been examined in Drosophila have an abbreviated S phase where regions of the genome are not replicated or are under-replicated. And there also are situations where an origin can repeatedly fire to give amplification of gene copy number above the overall ploidy of the tissue. So just keep this in mind while I review some aspects about eukaryotic DNA replication for you. So DNA replication in eukaryotes uses the same machinery all the way from yeast to humans. And sites at which DNA replication can initiate become marked by binding of this complex of proteins called the origin recognition complex. And it marks those sites as potential sites of initiation by recruiting a protein complex that has the capability of acting as a helicase, but is in an inactive helicase form. So what happens with initiation of DNA replication is the helicase becomes active to melt the DNA to generate the single-strand templates and to recruit the polymerases and the replication factors. And then the next stage of replication is bidirectional movement of these replication forks to complete replication of the genome. And so we're really beginning to understand a lot about how replication initiation is regulated. But really, in no eukaryote do we understand how replication forks are controlled. And the reason it's important to understand potential regulatory mechanisms for the replication forks is that it's essential to complete replication of the genome. And so in cases in which there's an impediment to replication fork progression, if that occurs adjacent to a region where an origin has not fired, that can generate incomplete replication, which will generate genome instability and can be a factor in progression into cancer. And in fact, this is likely to be the case because there's a phenomenon in humans called common fragile sites. So these are sites that are really conserved through the human population. And these are genomic regions that become vulnerable to breakage if DNA replication is perturbed. And you can see that in these fragile sites here shown broken in the metaphase spreads. So there's about 100 of these fragile sites. They're tissue specific, and they're associated with rearrangements in cancer cells. So we wanted to understand how can we follow and study replication fork progression. And these examples of differential replication give us powerful models. So in the heterochromatin is largely repressed for replication in the endocycle, and through the euchromatin there are domains where if we look at copy number relative to ploidy normalized to diploid embryonic cells, we can find these valleys of depressed copy number. And we showed that these are regions that don't have active origins, and so presumably on their boundaries there are replication forks. This gives us a molecular localization of where to look for replication forks. Now the converse kind of differential replication, which is so far found only in one tissue, in the ovarian follicle cells, where a given origin can repeatedly fire, giving rise to forks that move bidirectionally to give you a gradient of amplified increased gene copy number, gives us a chance to both look at origin regulation, but also to know where the forks are to study fork progression. So we decided to go after this, and I want to tell you about a fascinating protein called suppressor of underreplication. This is a protein and a gene and a protein that were discovered in Igor Zhimulev's lab, and they were discovered from this phenotype. So as I mentioned, heterochromatin is largely repressed for replication in endocycling cells, and that means when you look at a salivary gland spread, you're not seeing about a third of the arm of each chromosome. So the suppressor of underreplication mutant jumped out to the Zhimulev lab because what they found is that these regions now had extra DNA with new banding patterns. And so we looked at a more molecular level, looking at these euchromatic regions that are depressed for copy number. And what we found is that all of them were dependent on the wild-type function of SUUR for repression of replication in the 3 tissues that we looked at. Okay, so the wild-type function of this is to inhibit DNA replication. And Jared Nordman in my lab looked at the converse case of amplification, where again we have these gradients in the follicle cells of increased copy number. And he found that if you didn't have SUUR function, the gradient was twice as long, as if the forks were able to move farther. And indeed, if we overexpressed SUUR protein, now the gradients were really constricted. And there was no change and no effect on initiation, but rather an inhibition of replication fork progression. So this is a protein whose wild-type function is to be a dosage-dependent repressor of replication fork progression. And this raises the question, how does it act? One could think of it acting as forming a barrier beyond which forks cannot progress. Or one could think of it as a protein that perhaps gets onto forks and impedes their progression. And so we wanted a way to try to distinguish between these models and functions. And we were able to do that because of work from Brian Calvi and Alan Spradling's lab, where they looked cytologically in the ovarian follicle cells at the process of amplification. And what they found is that if they pulse-labeled these cells with a nucleotide analog, they could see the sites of amplification where replication was occurring by nucleotide incorporation. And this is because this happens after replication of the genome has been developmentally shut off. So what you start to see is that this incorporation goes from being a sphere to resolving to these double bar structures. And Julie Claycomb, a student in my lab, did a lot of work tracking this down, and what she found is there's control of the replication events in these cells such that there are specific windows in differentiation when origin firing happens, and then origin firing ceases. And if you pulse label at these stages, the sole sites at which you see nucleotide incorporation are the sites where the replication forks are. And so you see this resolution into these double bar structures. Here I'm just showing you one of the amplicons, where the width, the space between them marks the distance that the forks have moved away from each other, and the labeling marks the sites where the forks actually are. So I think you can see this gives us a very direct way to ask the question, how does SUR act? And so Jared, using an antibody, looked at SUR localization relative to nucleotide incorporation. And he found that when amplification first starts, that there's no SUR present at the replication forks. But then a little later, it becomes recruited to the forks. And it's very striking if you look when there's no more initiation and the forks have moved away from each other, you clearly see that SUR is at the forks. And he was able to confirm that SUR moves with the forks by chromatin immunoprecipitation. So here's what we think this protein does, that in a tissue and region-specific way, it can become recruited onto replication forks. And if those are in domains that lack replication origins, that can then lead to underreplication. In the absence of the SUR protein, forks born outside of the domain can cross over and replicate and restore replication. So Jared Nordman, who was a postdoc who did this work in my lab, now has his own lab at Vanderbilt University. And there's a bunch of interesting questions. Among them, how does SUR get recruited to replication forks? How does SUR inhibit replication forks? And could this be a mechanism in diploid cells to restrict replication of the genome to points late in S phase? And so Jared's lab, I'm certain, is going to answer all these questions. No pressure, Jared, wherever you are. But I'll encourage you to go to the poster from his lab on some of their recent really exciting results. Why is this important that there can be functions like SUR and potentially other proteins that can impede fork progression? The reason it's important is this can be a real source of contribution to genetic instability. And part of the reason I say this is from work that Brian Waugh did in my lab, where he found that there are zones within the euchromatin of the genome that lack origins, and that's constitutive across tissues, these zones that lack origins. But he found that that wasn't sufficient to repress replication, that although it was constitutive across tissues that these regions lacked origins, that they could be replicated in some tissues or not replicated in others. And what our results really strongly indicate is that layered on top of this constitutive establishment of origin-free regions is this second layer of regulation of controlling replication fork progression. So that in tissues in which there's no inhibition of fork progression, these regions can be replicated even though they lack origins. But if there's fork inhibition, that generates under-replicated regions that then can lead to genome instability. So there's one more aspect of genome instability from fork control that I want to tell you about tonight. And that is a proposal from the cancer field. So what researchers in the cancer field proposed is that there might actually be two reasons why gene amplification is detrimental. One being the increase in gene copy number, but the second being that if an origin refires and you have two forks on the same strand, if those forks collide, that can lead to double-stranded breaks. And so this is a hypothesis in the cancer field, but the limitation in mammalian cells is how to find the sites in the genome where you expect forks to collide to really test this hypothesis. And I hope I've convinced you that in these follicle cell amplicons, we know exactly where the forks are, and we know exactly where they're going to collide. And so what Jess Alexander in my lab realized is she could use that cytologically to test whether the sites where forks are predicted to collide, in fact, are sites where double-stranded breaks are formed. And she could do that cytologically by using these markers for double-stranded breaks, this modified histone, or these long tracks of RPA binding protein. And again, EDU marks where the forks are on the flanking the amplicons after there's no more initiation. And these markers for double-stranded breaks showed that indeed these sites have double-stranded breaks. So indeed, sites where you expect forks to collide do yield double-stranded breaks. And furthermore, Jess was able to use this system to ask, what does it take to repair these breaks, by asking what's required to allow the forks to resume progression. And surprisingly, what she found is the double-stranded breaks do get repaired, but they get repaired by non-homologous end joining. In other words, by a mutagenic process that further generates genome instability. So my lessons from this third vignette are that unusual biological processes that might seem like they're esoteric can be very powerful in providing experimental approaches and inroads to gain insights into fundamental problems in biology, in this case really uncovering aspects of regulation of fork progression that are important in human disease and cancer. And so we've learned that there's active inhibition of fork progression, and that indeed fork collisions from forks on the same strand can lead to genome instability. Collaborations also were very important for these studies. We've had a long-term collaboration with Steve Bell at MIT on initiation and ORC. We also collaborated with David McAlpine at Duke. We collaborated with Igor Zhemyulev's lab on SUUR, and the work on double-strand break repair was done in collaboration with Mitch McVeigh. So let me just close then by reiterating lessons that I found to be valuable in my research career. I still assert strongly that going after basic biology and foundational problems to yield critical, unexpected insights that have relevance to human disease and being able to pursue them in an unbiased way is important. And being able to apply a variety of approaches, including high throughput, can be critical. I mentioned community resources that were critical for these particular projects. They built on the foundation of so many of the resources that you heard about from Debbie Andrews, Flybase, Bloomington Stock Center. Again, that generous members of this community have really poured their hearts and resources into generating as tools for the community, and the value of collaborations. So the last thing I really want to leave the students and postdocs with is that Drosophila is an amazing model organism. There's so many exciting problems left to solve. I told you some of the questions that have come out of this work that need to be solved, but there's lots of interesting biology to solve, and this community provides you with the tools to be able to do it in a really efficient way. So you've landed in the right place, and I think you're going to have a great research career. The great thing about this job is the people you get to work with. It's been wonderful having students and postdocs and technicians come through my lab. Some of them are shown here. You may recognize some that have gone on to head Drosophila labs: Bruce Reed, Sharon Bickle, Andrea Page McCall. Many of these people worked on the side of the lab I didn't talk about tonight, looking at chromosome segregation mechanisms. In addition to these students and postdocs, we have benefited tremendously in the past few years from having Tony Mahowald in the lab. So I mentioned the contributors of the most recent work, but this is a list of the students and postdocs who contributed to the work on Pangu over the years. Boreana and Amir are here at the meeting. These are the students and postdocs who contributed to the Endo cycle work over the years, and Jessica von Stettina is here. And these are the students and postdocs who contributed to the work on amplification and replication for progression. All of this work was made possible by two long-term technicians in the lab, Raisa Fomina and Helena Kashevsky, and George Bell, the head of the Whitehead Bioinformatics Group, made many of our high-throughput studies possible. These are the agencies that have funded our work most recently, and thank you very much.